So, hi, welcome everybody. It's a pleasure today to host the seminar by Giacomo Mazzamuto. Giacomo is a researcher at CNR INO. Actually, the building that you see walking uphill towards the observatory on the right. And Giacomo is working in our department in Sesto Fiorentino, the campus that is close to the airport, and is part of the biophotonic group, which is a huge group of the department, and is running extremely nice experiments in, I would say, neuroscience. So they develop techniques to see the functioning of living brains, microscopic techniques. And I guess that today Giacomo will provide an introduction to the field, telling us something about the beautiful experiments they run in Sesto Fiorentino, and then of course the handling of the data that they produce through machine learning techniques. So I leave the floor to Giacomo, and of course, if you just have questions, you feel free to interrupt whenever you like.
Okay, thank you, Duccio, for the introduction, and good morning, everyone. It's great to be here in this beautiful hill of the historical headquarters of the physics department. And today I will give you a short introduction on high-resolution microscopy techniques for imaging of large biological tissues. I will show you that these techniques pose a number of implications in terms of the big data that we need to process, and this is why, in the end, we have to resort to some techniques also based on artificial neural networks or machine learning techniques. Before we start, I just want to show you a joke, a thought-provoking joke that I found on the internet. So the difference between machine learning and AI, if it's written in Python, it's machine learning. If it's written in PowerPoint, it's probably AI. Today, I'm doing PowerPoint. So this is just to say that I won't be doing a lecture on machine learning specifically. At the very last part of this presentation, I will show you some applications that we do. But first, I will give you a broad introduction of this scientific field, which is very interdisciplinary, as you will see in a moment. And I hope maybe not all of you are familiar with this kind of science. So I hope to give you a short introduction. So to slowly dive into this topic of microscopy, I just wanted to show you what things looked like back in the old days. Everything was done by hand. Everything was analog. Image acquisition, image processing, visualization and storage, everything was done by hand. Here you see Santiago Ramón y Cajal, who won the Nobel Prize together with Golgi, actually, for this kind of he was famous for this kind of illustrations that he did with his own hands. Nowadays, everything is, of course, digital. All the phases are digital, which is very nice and convenient, but it poses a number of problems in terms of visualization and storage, particularly because as I'm going to show you, these instruments produce huge amounts of data. This is a light sheet microscope. I will talk about this more in detail later. This is a commercial system. Of course, we can buy this kind of systems off the shelf, but we build and design our own systems, as I will show you in the next slides. And the problem with storage and visualization is so important that what we are trying to do is to actually swap these two parts, to swap image processing and storage, in the sense that we want image processing to happen as close as possible to acquisition, so that later we store or visualize only the processed data, because sometimes we can't really afford to keep all the data that we produce. So this is the direction where we are going. But I want to give you some numbers to put things into perspective. Let's say, before I give you some details about this kind of microscopy, Let's take a typical sample that we that we study in our lab almost on a daily basis. Let's take the whole brain of a mouse. This is roughly 1 cubic centimeter in volume. And if, let's say, we image at a resolution of 10 micron, three-dimensional resolution I'm talking about, so this is a voxel rather than a pixel, just one channel, that is to say one wavelength, one color, is 2 gigabytes in size, pretty manageable nowadays. 3 channels, 6 gigabytes, still pretty manageable. Of course, if we go down, if we increase resolution and we have a resolution of 1 micron or even less than that, like we are able to achieve in our lab, of course, everything scales with the volume, so we go to 2 terabytes in size for just one channel. Considering I'm considering a 16-bit pixel, so this is the dynamic range that we have, so 2 bytes per pixel. So we have a data set of 2 terabytes for just one channel, or 3. For 3 channels, we have 6 terabytes. So this is the range where we work right now, every day. So usually we launch our measurements, our acquisitions during the night, because everything is automated, so the acquisition goes on by itself. And the morning later, the next morning, we have some 6 or 10 terabytes of brand new data to handle. So this is the amount of data that we produce. And it is apparent already from here that it's important to have a processing pipeline that is efficient to be able to handle this stream of data every day. And this is all the more important if we ever want to scale things up to even bigger samples. I'm not saying that we will do a whole human brain with this kind of microscopy, but still, if we want to scale to even bigger samples, the data sizes explode pretty quickly. So you might have noticed that I'm talking about three-dimensional microscopy, because, of course, biological samples are inherently three-dimensional, but not the traditional microscopy techniques, in the sense that, yes, you can focus on different planes within the sample, but of course the out of focus light will render your image blurry at some point. So traditionally, what scientists and anatomopathologists, biologists have been doing is to physically cut the sample. They create thin slices of the sample that have a thickness that is comparable with the optical depth of the microscope. And so they physically cut it and they create a series of slices. So they physically cut the sample. Of course, this has some problems because it leads to sampling bias. You lose depth of information. You might damage the tissue. You might get visual artifacts. So the kind of microscopy techniques that I'm going to talk about today perform so-called optical sectioning. So the ability to be able to look in the sample at different planes within the sample. And I will show you how these techniques work and what they are based on. Let's talk about selective plane illumination microscopy. This is a term that encompasses a broad family of techniques that enable optical sectioning. So the ability to look inside the sample at different depths. These techniques are based, I like to say that these techniques are based on 3 pillars. First of all, optical clearing. I will show you in a moment what optical clearing is in the first short part of this presentation. Optical clearing is a biochemical process that you can use to make your sample completely transparent to light, sort of. This is a whole mouse brain before and after optical clearing. You can see that you can actually read through it. This is important because this light has to be able to penetrate deep into the sample for this kind of techniques to work. Then there is the actual instrument development part that we do also. And then finally, image processing, which is the area where I'm mostly active. I actually do more software and image processing rather than artificial neural networks, but some people that work with me also do these kind of things, and I will show you some results also in this respect. And you can already see how this is a very interdisciplinary kind of science, because you need the biochemists to put together and make these clearing protocols work. You need the physicists and optical engineers to actually build the microscopes, unless you want to buy them off the shelf, but they have some limitations, of course. And finally, there is a big part of image processing involved to be able to analyze this data. So let me give you a very, very, very quick primer on optical clearing, because this would need a dedicated seminar just for this. And also it's not my field of expertise, but I just want to give you an idea of what these techniques are. So why do we need optical clearing? We need optical clearing because biological samples are opaque to light. No light cannot enter into the sample, and this is not because of absorption. Actually, some tissues do absorb a bit of light, but it's mostly because of scattering. If you want to model a biological tissue, broadly speaking, you could think of it as an ensemble of particles having a relatively high refractive index of, let's say, 1.5, immersed in a watery medium with a lower refractive index of 1.33. And this mismatch between index of refraction makes light scatter, and that's why the sample is opaque to light. So a number of protocols have been developed to make the sample transparent to light. Either you decrease the refractive index of the surrounding medium, or you increase the refractive index of the particles. Anyways, there are many protocols. For example, you can use organic solvents that have a high refractive index, and so they match with the refractive index of the particles inside the sample. You can use aqueous solutions. These have a lower refractive index, so they achieve a lower clearing power, but they preserve the endogenous fluorescence of the sample better. Or these are techniques that became very popular in the last 10 years or so. These are chemical methods that are actually based on tissue transformation. This kind of technique was pioneered in 2013, so less than 10 years ago. The pioneering technique was called Clarity. This is a method for the transformation of tissue into a hybridized hydrogel form. So yes? What kind of tissue do we look at? Okay, we want to. No. We just want to get rid of those particles that actually make light scatter. That are mostly lipids. So you want to get rid of them, but these techniques do preserve the structure of the sample, the three-dimensional structure. So you can still see the individual neurons. After you do optical clearing, you just remove the lipids, the fatty parts mostly, but the structure stays there, and you can still see the neurons in 3 dimensions. And this particular technique goes even beyond that, because it actually sort of replaces the scaffold, the protein scaffold of the tissue with a hybridized hydrogel. So you see, it becomes sort of jelly like this kind of samples treated like this. But still, there are several techniques. We are able to reproduce all these techniques in our lab. Clarity, but also Switch is another technique that uses a different hydrogel, because Clarity uses this acrylamide nanoporous hydrogel. We use glutaraldehyde. So there are very different kinds of clearing techniques. But one nice thing to know is that these hydrogels are permeable. They have nanopores, and so you can also use this to, I mean, these clearing techniques are compatible with custom staining protocols, because as I will show you in a couple of slides. Clearing is a broad science. There are so many different techniques, different pros and cons. It depends also on the kind of tissue that you want to clear. For example, even brain tissue, it is different if you want to clear tissue coming from an adult brain or from a child brain. It's different because children have a different myelination level of myelin in the brain. And so every protocol, you get one protocol for your kind of microscopy or for your kind of sample, or even for the kind of fluorescent antibodies that you want to put into the sample. So not all of them are compatible with each other. Some have their pros and cons. This is a broad science. This is just to let you know that we also do research also in this field of optical clearing. So there is a whole classification of different protocols based on the technique, or on the optical technique, or on the tissue, and so on. And before we move to the next part, I want to show you, okay, this is, as I was saying, this, now, since the sample is completely transparent, then what do we look at? We look at fluorescence. So these are methods that work with fluorescence. This can be either endogenous fluorescence of proteins or molecules that are intrinsically within the sample and are fluorescent when excited with a laser light, or most of the times we do staining with fluorescent antibodies. These are examples of completely different antibodies targeting different parts within the cell, and or different structure within the tissue. It is important that the clearing protocols are also compatible with the staining protocols. And so it's important that these clearing protocols leave the sample permeable to these staining molecules, okay? Because they penetrate into the sample via diffusion mostly. So they have to be, if you make the sample transparent, but you leave it sealed so that antibodies cannot penetrate, then you can only look at endogenous fluorescence. So it is important that the sample is also penetrable to this kind of staining molecules. And to conclude this part, I just want to show you a slide that I think is impressive. These clearing techniques, especially those based on hydrogels, or these chemical protocols that hybridize the structure of the sample with this hydrogel, are also compatible with another kind of microscopy that is called expansion microscopy. What is expansion microscopy? Since, of course, you cannot increase the resolution below the diffraction limit, some people have thought, why not make the sample bigger? And so they have created expansion microscopy. Expansion microscopy is impressive. This is something that we do in our lab at Lens. So you see, this is a whole mouse brain before clearing and expansion. And this is the same brain after expansion. It increases by an order of magnitude. This is pretty impressive, I think. And it happens overnight. In just one night, it grows like, I don't know the exact workings of this protocol because I'm not a biochemist, but I think it's impressive. Of course, with these samples this big, we usually look at a smaller portion in the sample. Otherwise, the numbers that I gave you at the beginning are referred to this volume. Imagine, so this volume that is 10 times, one order of magnitude bigger. Yes? There might be a distortion, but mostly are maintained. Yes, you see the structures that are essentially magnified. Instead of magnifying optically, you make the sample bigger. I don't know the details of these protocols. I don't know. It might be a polymerization thing or something like that. But once you have this hydrogel, I don't know exactly how it works. What are the steps? If it's steps in temperature or a bath, you have to put the sample into some baths or something like that. But they are able to. This was done by our PhD student Neve Brady. So this is also one thing that we are starting to do right now. So this is just to give you an idea of what crazy things people are doing around the world. No, I don't think it's feasible because these things basically work by diffusion anyways. So whatever you put your sample to whatever bath, the molecules or the chemical need to be able to penetrate into the sample. So now I'm sorry if I can't be more specific, but it's not really my expertise. Yeah. No.
But because it's not the same, you have no proof that you didn't make any limitation because modifying the original instance you are getting the wrong information.
But well, let's say we are doing mostly cell counting things, or we're not interested in a perfect, but it's I think it's still yeah. Yes. We have just started doing this. It's a new item in our list of things that we can do. I just wanted, this is a picture that I grabbed at the very last moment, but I think it was nice to show to people because me myself I was impressed with this thing when you see it live. I mean, anyways. So this is what people did. Instead of increased resolution, let's make the sample bigger. Anyways, now we move to the second part. I will show you some details about the actual instruments, the techniques that we use to do optical sectioning. So we've seen that these techniques have to be are based on samples that are transparent, right? Because otherwise light cannot enter into the sample, and fluorescence light cannot reach our detectors. What is the simplest form of instrument that has optical sectioning capabilities? The simplest is laser scanning confocal microscopy. It's a very simple system. You have a laser source that excites fluorescence on a plane within the sample, and then the fluorescence light is collected by a photomultiplier, a photo detector. You see that we have a pinhole here. This pinhole acts as a shield. It basically blocks the fluorescence light originating from planes that are not in focus, and this increases resolution and contrast. It's called confocal because these points A, B, and C are mutually confocal. So it's a simple system, but it's a point-by-point system. So you have to scan this point on the sample. So you need, for example, galvanometric mirrors. These are mirrors that you can scan quickly on the surface of the sample. Or another technique that we use is two-photon fluorescence microscopy. So we use two-photon absorption, which is a nonlinear process, and so it happens only at where two-photon absorption happens only at the plane where your laser is focused, and also you are pretty sure that the fluorescence comes from exactly there. So it has intrinsic optical sectioning capabilities. And but I just wanted to mention just for the sake of completeness, but I would like to spend some more words, a few more words on light sheet microscopy, which is what we do mostly in our lab. Light sheet microscopy is a microscopy technique that achieves optical sectioning using a sheet of light. So you create a sheet of light. How? For example, the simplest way of creating a sheet of light is to use a cylindrical lens. A cylindrical lens, like in this case, focuses only in one direction, but not on the other one. So you have a sort of plane of light, mostly at the region of the sample, and then you collect fluorescence from the top or from the side. It depends on the geometry. You can buy these instruments, but as I was saying at the beginning, we usually build our own systems that allow us to high degree of customization and adapt to the different kinds of samples. Several geometries are possible. Here you see the cylindrical lens. This is the simplest way of creating a light sheet. So you focus just along one direction, not the other one. So you get a thin sheet of light, ideally, and it's not as simple as in the theoretical world. Several geometries are possible. One geometry that we also use, and I will show you later, is this geometry where the objectives are tilted. That has some advantages, but also creates some problems in the processing part, because here the reference frame of the objective is not the same as the sample, so you have to manipulate the data, as I will show you later. Now I just give you a quick overview of what can be done with this kind of microscopy. First of all, they can be used also on live animals, live samples. For example, in this case, this is a larva of zebrafish alive. In this case, we don't have to do optical clearing because these animals are completely transparent up to 3-4 days after conception. After 3, 4, 5 days, not more. After that, they're not transparent anymore. But I think there are also requirements by the law that don't allow you to make experiments after a certain amounts of days after the conception. So, but at 2, 3, 5 days, the nervous system is already developed, and you can actually see it. And you can do fluorescence measurement on functional imaging. So you can actually see what areas of the brain activate when the larva is subject to stimuli such as a sudden flash of light or things like that. This also we do, not me personally, but there is a subgroup in our lab doing this. Or this is another famous study. People have imaged the development of a Drosophila larva. And so you can see as the larva, you can clearly see as the larva goes through the different stages of embryonic development. Now it unfolds, as you can see. This is also one famous result done with light sheet microscopy. I'm just showing you a list of examples. And this is a whole mouse brain. This is actually what we did in our lab. Perhaps later we have images that are more stunning than this one, but you can see the structures inside the mouse brain. Or this is the cerebellum. So you just the cerebellum part of that brain, and from this animation we can clearly see the optical sectioning capabilities. So we can do like a CAT scan, but just using light. There's no magnetic fields going on here. But you have a tomography, so just like a CAT scan. Or this is something that also we did in our lab. So it depends on what you want to target, what kind of fluorescence you manage to put into the sample. You can target blood vessels. This is the vasculature network. You can target a microglia. Of course, you can then superimpose the two signals if you like. But perhaps this is the most, it lags a little bit, not on my display, but anyways, this is the whole vasculature network of a mouse brain that we reconstructed with this light sheet microscopy, and we can go down and see down to the smallest capillary. One thing to know, yes? Let's say 1 micron or 1 micron diameter, yes, I would say. One thing to know about light sheet microscopy is that resolution is usually not isotropic, because you get high resolution on the XY plane. Let's say if, you're looking from a top and you have a high resolution along the XY plane. You have a worse resolution along the thickness of the sample, and that's because simply the light sheet has a finite thickness, and so it ends up still illuminating also other planes. But there are some tricks to make this problem less important. Optical engineers do a lot. You can do beam shaping. You can try different shapes of the excitation beam that have different profiles. And so depending on what, there is also a whole optical engineering part for this kind of light sheet engineering, let's say. But the fact that resolution is not isotropic also is a slight complication also in the image post-processing, because the final image that you get is not isotropic. As I was saying, there are several techniques to create a light sheet. The simplest is a static light sheet, as the one created by, for example, a cylindrical lens. So this is the light sheet, and this is the fluorescence that you collect through the objective. This grid that you see here is like the projection of the CCD sensor on top of the well. But one thing that we do in our lab is to use a digitally scanned light sheet microscopy. So we just use a laser beam, and then we scan it quickly along the sample, and this creates sort of a light sheet, as I will show you in a moment. Light sheet microscopy can also be confocal, like the system that I showed you at the beginning. This helps in eliminating the light coming from planes that are not in focus. And you can see here that there is a slit. There is a slit in the detection path that acts as the pinhole that I showed you at the beginning. So it blocks light coming from planes that are not in focus. Here you can see it in this animation. There is a galvo mirror here that scans the light sheet in the sample. There is a second mirror here that makes the fluorescence light go through the slit, and the third mirror that does so-called descanning. So it makes the fluorescent light impinge on different parts of the sensor on the CCD camera. So this is confocal light sheet. It helps a lot in increasing contrast and resolution, but it requires a lot of moving parts. You see, we have 3 mirrors here. So one thing that we do, we not only do digital scanning of the light sheet, but we use these cameras that we use, because manufacturers have been improving their products also to solve the problems of light sheet microscopy. So the cameras that we use have a mode of operation that is called rolling shutter. In this mode of operation, instead of exposing the lines of the sample all at once, we just expose each line at a time. Okay, so each line is exposed at a time. And of course, you have to make sure that everything is perfectly synchronized. Only the line of the sensor where the light sheet is impinging at that moment has to be exposed. So everything has to be perfectly nice. Otherwise, you just see black, of course, right? So if the line sheet is here, but you are exposing this line here, you always see just a black field throughout your acquisition. So you have to make sure that everything is synchronized. So there is some electronics to drive all this system. A global shutter, that's because you will end up collecting light from portions of the sample that are not, maybe at some point you see this later in this video, where I'm starting the video from the beginning. So this is the light sheet scanned on the sample. Instead of using global shutter, we expose one line at a time. And what happens, you will see it in a moment, is that basically you throw away, because the light sheet would actually excite more than one line, right? So you basically throw away the contributions from the nearby lines that would be excited. There is still some light, sorry. You still get some light impinging on the nearby lines, but those are not exposed. So it actually works as a slit. It just works as a slit, but it's not a physical slit. It's like this trick of actually exposing only the line on the sensor that is illuminated at the moment. So everything has to be perfectly synchronized, and it's not a terrible thing to do, but requires some electronics and cameras that support this kind of operation. Before we move to the data analysis part, I just want to give you an example of the instruments that we build ourselves. So we actually do the optical development. These are two different setups. This is a light sheet having the inverted and inclined objectives, like the geometry that I was showing you a moment ago. And this is a different setup that has while this setup here has a big and wide chamber where we can actually place even 5 or more samples. This setup here with a chamber that is very wide is useful to image samples that are thin but large, like slices of human brain, as I will show you in a moment. Whereas this setup here has a chamber that is bigger in volume, and so it is mostly useful for samples that are bulky in a sense. And this is a whole mouse brain, again, after optical clearing. We put this cuvette into this chamber that is then filled with an index matching liquid, and you do the imaging. So we developed both of the systems. Here you see that the sample chamber is here. These are the two objectives, 1 and 2. We have 4 laser sources. We can image 2 colors at a time. So remember those numbers that I showed at the beginning, you have to multiply by 4 because we have 4 wavelengths. Of course, each wavelength excites a different fluorophore that targets different parts of the cell, of the biological structures. We are able, for example, to distinguish excitatory neurons from inhibitory neurons. They fluoresce in a different way, so we are able to see this. Data acquisition and control is not trivial. So we had to develop not only the hardware, but also the software for data acquisition. This is not an important part, but I wanted to show you because I think it's non-trivial. I mean, this system has to sustain a data rate that is around 1 gigabyte per second, mostly 1 gigabyte per second. And it has to sustain this rate for several hours because the acquisition lasts several hours. So we have to use solid state drives. We have to do memory management very precisely. And so we had to develop our own software. You can find it on GitHub in case anyone is interested. And everything, it's written in C++, runs on Ubuntu, which is nice because it has a very nice uptime. You don't want your system to start a system upgrade while you are in an acquisition, especially at night when it usually happens. And so we're happy about this. Another thing that we need to do in our processing pipeline is so-called image reslicing. This has to be done only when we use the microscope with the two objectives that are tilted. And this is because the objective is tilted at 45 degrees relative to the ground, but the sample is moving. There is a translational stage that makes the sample move. The sample is moving horizontally. So you have to apply a coordinate transformation, which is actually an affine transfer to go from the objective reference frame to the lab reference frame. So in the end, you get the final shape of the process sample would be like this, because the objective is imaging like this, okay? It's not a complicated operation to be done, but it has to be done on data that are this big. So even the simplest of the processing steps can be a problem when you have to do it on terabytes of data. And slowly moving on to the data processing, I want to show you some acquisition campaigns that we did. This is a whole human hippocampus that we got from a donor. Okay, some samples that we get are, of course, if we use animals, the animal is sacrificed. We also get some samples from surgery, as I will show you later. In this case, of course, it comes from a donor, so from someone who decided to donate their body to science. And this is the whole hippocampus that was imaged using the inverted light sheet microscope, because it is useful for samples that are for large sections. Of course, we had to cut the sample anyways. We had to cut it, but still the sections that we make have a finite thickness of 1 millimeter or 0.5 millimeter. So we can still do three-dimensional imaging of the slice, even if in some cases we have to cut the sample, but not, for example, with the mouse brain. The mouse brain is intact. Or we also do the whole mouse heart, for example, an intact and entire mouse heart. We've done it. So without cutting the organ. In this campaign was for a total data size of 24 terabytes. Not much, actually. I will show you later something that is even bigger. This data is also online. It can be downloaded, although at a lower resolution. This was done in the Human Brain Project, a project by the EU where we have been part since several years ago. This is a bigger challenge. This is the big portion of a human brain, and specifically the Broca's area, which is, I think, something related to language, if I can remember. And this was a massive undertaking because it had to be cut in 48 slices. Each slice had to go through optical clearing. So this is the slice before and after optical clearing. And this is what we get. So you see the slice before and after optical clearing. And this is the result of imaging with the inverted light sheet microscope. Again, because these are several centimeters in size, you see, 4 centimeters by 4 centimeters. So you need a sample chamber that is able to accommodate this kind of samples. And the thickness is 1 millimeter or 800 microns or something like that. These are the 4 wavelengths. We target different cellular structure or different cells. In this case, we are targeting the neurons, and you can clearly see that. Just to give you an idea of how big this image is, you can zoom in several times, and you can see, we can clearly distinguish the individual neurons. And this is where then segmentation and machine learning comes in, as I will show you in a moment. But we can clearly see the gray matter, the white matter, the gyri of the cortex. This was done in a project that we have together with the National Institutes of Health and the Massachusetts General Hospital in the US, that they actually gave us the sample. So it is a slice. We had to cut it at some point, but it is a three-dimensional slice. So we can look into the slice at different planes. This campaign here was done in just 1 month, in the month of August last year. In just a month, we acquired 48 slices, 4 channels each, 500 terabytes. So half a petabyte of data came out of the microscope. We can't keep it all. It's impossible. So we apply lossy compression. We use JPEG 2000. JPEG 2000 is a compression algorithm. Perhaps there's a slice later. And we go down from 500 to 25 terabytes. So we throw away some information. These are lossy algorithms, okay? Like MP3, for example. MP3 throws away those frequencies that mostly we can't hear. So at the end, the visual result is still pretty good, even though the data set size is much smaller. So basically what we throw away is noise or background, basically. Then we have to do reslicing. We also do downsampling at some points because we have to upload the data back to the US because the projects ask us to share the data with the public, but we share the data at a lower resolution that is already, however, 10 terabytes that I had to upload to the US. Thankfully, we have a 1 gigabit internet connection at Lens, otherwise it would be impossible to upload this data, not download. Anyways, and now let's finally move to the big data management part. I will show you some challenges that arise in dealing with this kind of data sets, and also some application of artificial neural networks for automatic cell segmentation, cell counting, and things like that. So our pipeline might seem complicated, and it is. We do light sheet microscopy, we do 2-photo microscopy. We have different streams of data. For example, we can have grayscale images, color images, different kinds of resolutions. The first thing we need to do is image stitching. I will spare just a few words on this. Image stitching in 3 dimensions. We do data compression, both lossless and lossy. We do downsampling when needed, and we train artificial neural networks using manually annotated data. And then we do, for example, neuron counting, neuron classification. I will show you just a couple of examples. This is a whole mouse brain that we acquired at our microscope. You see the tomography. So as time goes by, there's no time here. We're looking at different coordinates along the thickness of the sample, okay? This particular data set was 4 terabytes, but 4 terabytes don't come in a single file. It's a grid, it's a mosaic. In this case, it was 15 by 12 stacks, overlapping stacks. Each stack in this particular case was 23 gigabytes, but it can be more than this. Each stack has 3,000 frames. So we have, first of all, we have to fuse these stacks. There is an overlap between adjacent stacks. Otherwise, it would be impossible to create the whole tomography because, of course, the microscope has a limited field of view. So it just sees a small portions of the sample. Then we move the sample and acquire another stack. So everything has to be fused together in 3 dimensions, which is a challenge per se, but it's even more of a challenge if the data is 4 terabytes. There are some solutions, but at the end, we had to develop our own. And this is available also on GitHub. It's a software that I made. It's called Zeta Stitcher. And what it does, it doesn't do miracles. First of all, the basic assumption is that the microscope works well. So it doesn't do correction of aberrations or distortions. So the assumption is that the instrument works well, but still you have to compute the best alignment between adjacent stacks. It does some approximations, by the way, so I don't read the whole data set, but just do a sampling in the data set, so it's pretty fast. And one nice thing that we have is that I said there was no Python, but there is some Python in this presentation. We have developed this API that allows me to access arbitrary portions of the virtual tomography without actually having to produce the tomography. Otherwise, I would have to produce, starting from a data set that is 4 terabytes, but divided in tiles. If I were to produce the tomography, I would create another 4 terabytes of data. So instead, we use a virtual approach. So we leave the data set as it is in tiles, and then there is a simple array interface that behind the scenes takes all the chunks from the dataset and creates and gives you the volume that you ask. So if you ask for this volume, it will take this part from one tile, this part from another tile, this part from another tile, and I will give you the volume. It's very convenient, especially if you want to feed a data set that is this big to, for example, an artificial neural network. Of course, you have to feed it in chunks, in small chunks. And you see that we can actually see the individual neurons. These are the cell bodies of individual neurons. Yeah, compression is a big subject, it's a big topic. We tried different algorithms. At some point, we even tried video compression algorithms, like MP4, for example, because our tomographies look like a movie in a sense, because consecutive planes are very similar. And so we thought, why not try video compression algorithms that should take advantage of the similarities of consecutive planes. These have some, it works, but it has some problems. At the moment, we are using JPEG 2000 with a compression factor of 20, actually, not 10, of 20 here. This is what we use. And in the end, this is actually a different algorithm, but what I want to convey is that in the end, these algorithms are lossy, but the visual quality is preserved. You see what basically, I don't know if it can be seen from there, but basically what is lost is the noisy part. So the structural parts are preserved. So we are, for the moment, happy with this. The reason why we use JPEG 2000 is because it supports lossy compression and 16 bits of dynamic range, 16-bit grayscale, which is what we need. And for the moment, I wasn't able to find alternatives that support lossy and 16 bits. These two things are hard to find together. But maybe in the future, we will move to different algorithms. I don't know. And now, finally, I'm going to show you some segmentation applications. This is a piece of human brain cortex. Human brain. This comes from surgery. Before and after optical clearing. In this case, we used the 2-photon microscope, not light sheet in this particular example. And this is the 3-dimensional reconstruction of this sample here. You see that as time goes by, we are looking at different planes within the sample, and we can clearly see the individual neurons. So what we want to do is to count these neurons automatically, because otherwise, especially for big data sets, it's impossible to count them manually, of course. To do this, we use a simple artificial neural network. I will show you some details, but very broadly speaking, we first do manual annotation because we have to train the neural network. So we draw the contours of the neurons, optionally placing them in different classes, if you like. In this case, for example, the axon was placed in a different class, so the network should learn to segment just the cellular body and not the axon, which is what we wanted to do. Then the network is basically a classifier in the sense that it classifies each pixel. Okay, so each pixel is classified based on what? If you consider that pixel to be the center of a patch, you ask the network, what is the probability for this patch to contain the visual pattern of a neuron? And then you assign the probability to that pixel. So you get a probability heat map. So the network doesn't actually do segmentation. It does pixel classification. So each pixel is classified. And then on the heat map, it's easier to apply standard segmentation algorithms, mean shift or something like that, to actually segment the neurons. Perhaps it's better seen here, so we can do the segmentation. And you see now that the contrast decreases as we go deep into the sample, but the network, I would have to readjust the brightness level, but the network is still able to see the neurons even when with this level of contrast we find it hard to see them. So it's a simple convolutional neural network, nothing fancy. This work was done in cooperation with a company that works with us. It's called Bioretics. It's a company, a small company, specializing in computer vision and machine learning. What I want to tell you about this network is that it is not a three-dimensional network. It's a 2D network. So each stack is processed separately, independently. But then, in a sense, if we then put the predictions on top of each other, in a sense, we have a three-dimensional prediction. We call this 2.5D because it's not 2D, it's not 3D. And but still, we are able to reconstruct the volumes. And so we can do what we call three-dimensional digital histology. So, for example, here we have colored neurons in different colors depending on their size, for example. This classification wasn't done by the network, was done by thresholding, basically. But still, we can distinguish small neurons from bigger ones. Maybe it's best seen in the next slide. This was actually part of a broader study where we did 4 samples. These 4 samples came from children, from the Children's Hospital here in Florence. And we did this cell counting. And for example, we can plot the cell counts, we can plot the mean volume. And you see that we can see some layers here. These are actually the layers of the cortex, because you see here, there are some layers. There are some layers where there are small and sparse neurons. There are other layers where you have big neurons that are mixed with smaller neurons. And it's basically, you can see it here. So you can see that we can do in 3-dimensional reconstruction, you see. We can go into the sample. Because the cortex is actually organized in layers. There are different layers that have neurons with different characteristics. Some layers have bigger neurons, some layers have smaller neurons. And since it's not super high, I mean, compared to the standards of machine learning, it's not super high, but I know it might be 80% or something like that, or 85, it depends. But still, it's considering the size of this data, it's still something to work on. And we can see these layers. We can see these layers, and for example, since some of these subjects had some pathologies, apparently some of these pathologies alters the way that neurons are distributed along the layers of the cortex, and so we can more quantitatively study this kind of things. If we go back to the same sample that I showed you earlier, the big acquisition of the of the broadcast area of the human brain cortex. As I was saying earlier, we can see the neurons here. This is light sheet microscopy. And there is a PhD student of ours, Filippo Castelli, who designed a unit, basically. But it's the same principle of the network that I showed you earlier. It's a two-dimensional network again, and then we put the predictions on top of each other. But in this case, it's a unit rather than a standard convolutional neural network. And it kind of works. This is the raw image as it comes out of the microscope. This is the prediction done by the network, and then finally we do instance segmentation to distinguish a neuron from the nearby neuron. And you can see it here as well in 3 dimensions. You see that the shape of the sample is like a trapezoid, or I don't know how to say it. That's because it has been processed to correct to go from the objective reference frame to the plane to the sample reference frame. It's the transformation that I was showing you at a few moments ago. So you see, it's like this. 2 more examples, and then we are done. This is a work that was at the very beginning done in cooperation with the Department of Information Engineering here in Florence. In this case, we are working on a mouse brain. And they developed a neural network for image enhancement. So the network doesn't do segmentation itself. So the network is trained to go from the actual image taken at the microscope to an enhanced image, where ideally it acts like a filter, basically, where you discard all the features or fibers that you don't care about, and you are left with ideally just blobs in a black background, and then you apply a standard localization algorithm to find the centroid of these blobs. So there is again a training phase where a human has to go there with a lot of patience and put a centroid and put a marker at the centroid of every blob from the image coming from the microscope. So we have just a text file of coordinates, X, Y, and Z. These coordinates are used to create a synthetic image. So an image created just with a black background and blobs placed where the neurons are supposed to be. So the network just learns to transform the patch coming from the microscope to the ideal patch. So it learns to just clean the image. It provides a better image, an enhanced version of the image. Then in prediction, of course, we feed the network with a patch that the network has never seen, and it should produce an image where you have a higher contrast between the background and the blobs. And finally, we use a mean shift algorithm to just find the centroid of these blobs. Yeah, so ideally the network just transformed this blurry input image to an ideal image where the blob is more evident. And for example, this is the result applied to the whole mouse brain. In this case, these are 2 million neurons that were automatically localized. And once you have this, then you can do numerical studies. For example, you can count how many cells there are in different parts of the brains, because we do the alignment with the reference atlas. There is a reference atlas of the mouse brain, so we can count how many cells there are in different areas of the brain. We can do, for example, cell density, how many cells per unit of volume. We can plot the clustering, 3-dimensional clustering index, the tendency of cells to aggregate together in clusters. So we can do digital histology in a sense. And this is better seen in 3 dimensions. So we are able to do this kind of studies in 3 dimensions. This is again the clustering index. And this is it, basically. I just want to show you one last slide. You might have heard about DALL-E, which is this very popular artificial intelligence platform developed by OpenAI that is very popular nowadays because you can generate an image starting from a text prompt. And it's everywhere on the internet, on social networks. People are becoming so-called prompt artists. This is a new job for the future. I was on the waiting list for several months, and then they gave me access. Now the waiting list is just 1 day. I think you can get an access in 1 day. And so I tried to enter this input. Neuroscientists finally discover how the human brain works. And this is what DALL-E produced. These 4 images, which I think it's pretty stunning. DALL-E still has some problems with faces. They're not perfect, but still, I think it's pretty impressive as an output from an artificial intelligence software. So I just wanted to end with this nice pictures, because it's super popular nowadays. In these very months, everybody's talking about this thing on the internet. So I just wanted to try it. And I think that this network, and it's not a network, these are diffusive models. I don't know exactly how it works, but they have learned not only the world around us in terms of objects and things, they also have learned like sort of physics. They know, look at the reflection of this light on the face. Or for example, there are other images where you see how light propagates in crystal structures, and it's perfect. I mean, it's as if it knows physics also. Or there are other pictures where it is clear that the network has clearly a sense of gravity, for example, that we live in a world where we have a privileged direction that is dictated by gravity. There are some images where this is very, very apparent. And so this is it. I know that I had to go very fast. This is again the SST positive neurons in the whole mouse brain. I had to go very fast, but I mean, the aim of this presentation was to give you a very broad and quick introduction to this kind of very interdisciplinary science. So we've seen clearing, we've seen optical development, a bit of image and data processing. It wasn't my intention to be very detailed. I just hope you learned something new, something hopefully interesting, and that's it. Thanks. Of course, I'm open to questions or curiosities, if I'm able to answer.
Yeah, thanks a lot, Giacomo. Very nice and very inspiring. So do you have questions for Giacomo? Or remarks? Yeah.
How often?
The question for the recording is how often do you do the scans, and like how much data you already have and how much you expect to be getting in the future?
I would say that in some periods, we do a mouse brain almost per day. And so this should give you a number of how many brains we deal with, many. Because, for example, some colleagues of mine are doing studies on fear memory, how the memory of fear develops. And so they have mouse that are trained to fear, for example, a particular portion of their cage because they get a small shock. I don't know all the details about this, but they are able to, with an injection of a tamoxifen, I think, where they inject tamoxifen into the mice, and they are able to sort of block the fluorescence of the activated neurons at that very moment. And then after that, the brain is imaged with this technique. And in principle, we should be able to see which neurons were firing up during the experience of this aversion, the creation of this aversive memory of fear, for example. There are other studies that have more of an anatomical goal, like the ones that we do for the NIH, the National Institutes of Health, the big study of the human cortex. Those are mostly more than a scientific at this moment. They are a technological challenge more than a scientific one. It's the equivalent of the genome mapping. You know, at the beginning, mapping the genome was just a technological challenge. Then, of course, it developed into a field. Now, mapping the connectomics in the whole human brain is, first of all, a technological challenge, and then it will, for sure, deliver some scientific results. So we are at this stage for the moment. In that case, in just 1 month, we produced 500 terabytes of raw data. Then we compressed it, but that happened just 1 month. Of course, we're not doing these kind of acquisitions every month, but 2, 3, 4 times per year, yes. So I am afraid that we produced several, I don't know, 5, 6, 10 petabytes of data from the very beginning, raw data. Then we have a storage of 1 petabyte, not more. 500 terabytes are here. We are always growing. Maybe this is our computing cluster architecture. We have a small computing cluster with a job submission system where users can send their jobs and queue them, put them in a queue. It's convenient because the job might run at night when the slots are available. We have GPU servers. We have like 10 or so GPUs. We have a 10 gigabit private network. It runs in parallel with the network of the institute, because we really need 10 gigabits per second because the data are huge. We store some of the data to Cineca. We have a storage there also, and we have a fiber, dedicated fiber to Bologna, 10 gigabits. Otherwise, it would be impossible. We also have a decent upload internet, which is useful for sharing this data with the US. The repositories that we are required to use for this project are in the US, so I have to upload all the data. So every week there is a processing pipeline going on.
When do you think it will be possible to track down connections in the human brain, like, or if?
There are some people that are already tracking synapses, but on a limited number of neurons. I mean, not in the whole brain. Now, in a limited number of neurons, it's already possible. People are tracking manually or using automated techniques, are tracking single synapses. Some of them can be very long, centimeters also. They can go from one side to the other one. So the problem with neurons is that I can't remember how many neurons are there in the human brain, but billions. And synapses are a power of this number, so it explodes very quickly. I think it's possible to map the individual neurons in the whole mouse brain. It's possible. It's like 50 or 60 million neurons, which seems a lot, but it's not. I mean, if you were to place the coordinates X, Y, and Z in a text file or a binary file, it would be 100 megabytes. Every neuron in the whole mouse brain, ideally. 60 million, it's not that much. The whole connectomics of the human brain is harder, I think. It's being done, but on smaller. For example, there are papers where they track like 20, 50 synapses. And that's it.
Questions, comments?
So let me try to carefully ask this question. And I mean it in the kindest way possible.
Sure.
But think of it like, I serve on the NIH studies section of proposals like this. The debates are rough amongst the people on the panel, so I'm going to let it erupt on you a little bit. Why, it happens in projects where people build out all this capability, image all this high resolution, and all the computational stuff, and network diagrams. And it's impressive, expensive, and then somewhere in the project, somebody says, well, I'm going to use JPEG 2000 to impress it. And it was kind of disconnect when you read a proposal like this. Where you're like, why did we just spend millions of dollars so that we could use a human standard compression engine, not even a scientific compression engine, a compression engine used to make sure humans don't think the edges are missing things, right? That's what JPEG 2000 do. They were built to make it so humans can build images, not scientific data. There's a big difference between scientific compression versus
True.
treating the human thinking the image doesn't work.
True.
And then we do all this, and then we segment neurons that have already been tagged by 4 pores, which should be the first most simplest computer segmentation ever. Tag them with a frequency dependent tag that says you have no neuron. And we do it all, we can scan at high resolution, but in the end it's just chunked into, at best, you can cells, which are more than sort of 50 micron-ish size objects, right?
How much is
50 micron like size.
50 micron seems big, maybe less than that.
No, so
Maybe, I don't know.
bacteria would be 2 microns.
Anyways.
I'll give it 10 microns.
Okay.
That's like a blood cell. A blood cell is 7. So you feel the disconnect, that we made something, but it's like it's hard to take this and have, and the group I work with does the same sin. But you create all this data, and most of it doesn't matter because it's like you don't have. It's hard to squeeze scientific information out of it. The end, you're going to say, let's say I need something, right? And I didn't need to really kill myself for massive data sets at high resolution to do that. So how do we work through this conundrum?
That's yeah, understand what you mean. Speaking of JPEG 2000, I'm not super happy with the solution. We're looking into alternatives. Yeah, it's true that it's made for the human eye. That's true. But then the network sort of has to see these features. So one problem is if you train the network of uncompressed data and then you do prediction of uncompressed data, that's a problem. We've seen that if we use both compressed or uncompressed data and we train the network with the same kind of data set, it sort of works. So yes. No, it's easy, but the problems arise when the data sets are terabytes and terabytes of data, probably. Yeah, that's true, but there's a lot of variability across the sample. Maybe the threshold would need to change across the sample. You have other structures, you have fibers, you have other structures that you don't care about. Maybe that's where the network, the predictive power of the neural network comes in. It's
scientifically you get out of collecting events?
two examples come to my mind. For example, at the moment, as I was saying, this particular project with the NIH has more of a technological footprint rather than a scientific one, as I was saying in the presentation. I'm aware of this. Other projects also investigate, for example, how, for example, the children, these samples here come from children with different pathologies, degenerative pathologies. And we can see, for example, how the pathologies affect the cortex. Where are they? Here. This in the, sorry?
What is the neuronal density?
There are pathologies where the structure of the layers of the network, because the cortex is organized in layers. So I've learned, because I also didn't know. And layers have different characteristics, different features, and apparently some degenerative disorders change these layers. And we are able to see more numerically, let's say, how this happens in the we can count volume, density, so we can see the layers, and we can also see differences between the pathologies. So in one sense, there is sort of scientific quest. In the other case, this mapping here, this study that we did, this one, this is a more of a technological challenge rather than a scientific one. Two examples come to my mind. For example, as I was saying earlier, the human genome project at the beginning, it was a technological challenge to map the whole genome of the human, of a human genome. Then it created a new revolution in genetics. And this is the stage where we are at with this kind of studies. We're just pushing the technology, which has a number of consequences, maybe not related to this field, like when the technology to go to the moon was developed, then what are it had a lot of consequences for the general population and technology, even though there is no direct advantage of being gone to the moon. There is a lot of technological pushing here, more than scientific one. I'm aware of that. Yes. It's a promise for the future, but it's pushing evolution in a number of areas. For example, file formats. We are in the process of converting these huge. Yes. For example, there is a new format that is. For example, we are in the process of converting this data set in a format that is suitable for the cloud, for example, and with, of course, pyramidal resolutions. So, for example, there is NeuroGlancer. For those who don't know it, NeuroGlancer is a product by Google. So if you go to github/google/NeuroGlancer, so Google has developed a visualizer that works as Google Maps. It loads smaller chunks of your data in 3 dimensions, so you can see the 3 projections. And so, for example, we are putting our, because the files that we acquired are arranged in a sort of internal system that we came up with. We are, I'm speaking about file names or file formats, compression. We are in these days in the process of converting our data sets to a format that is suitable for NeuroGlancer. A NeuroGlancer is a way for people to navigate the data set in 3 dimensions. But in order to do this, And in order to take advantage of the parallelization that is enabled by the cloud, for example, one thing is to store your data set in a pyramidal format that is in the same file, like a H5, for example. Another thing is to store your data set in a cloud-friendly file system. So, for example, the ZAR is a format, I don't know if some of you are familiar with that, that you can use to store not in a single file, but in a folder that has to be organized in a particular way. And then you can put this folder in an S3 bucket. You know what an S3 bucket is from Amazon Web Services? It's a type of cloud storage. So some people are actually developing, also within the NIH, are developing new file formats that allow volumetric three-dimensional data to be stored on the cloud, on storage buckets like S3 from Amazon Web Services. And it's a different kind of storage than having a 100 gigabyte file with different resolutions embedded in the file. So there are still some areas where this thing is pushing the line in many, also unrelated, if you wish, areas. You could use the same kind of storage format for satellite imagery, for example, or for imagery coming from satellites, for I don't know, high resolution Earth observation data. There is still some besides the technological challenge. There are some areas where people working in this area, in these subjects, are trying to push the line forward, let's say. Then I agree with you. There is a lot of technological challenges here. Yeah, fundamental research, yes, is also one thing, but it's mostly done by the users of our technology rather than what we do ourselves. We also do some fundamental research in the sense of studying degenerative pathologies, but sì. There was a nice image that I probably should include in this presentation. Unfortunately, I don't have it here with me, but we did, for example, a three-dimensional section of three-dimensional portion of a human bladder, and that we did with light sheet microscopy. And we also applied a color transformation, a simple color transformation algorithm to make the acquisition look like as if it were stained with the traditional pink and violet stain, you know, hematoxylin eosin. This kind of stain here is what anatomopathologists are used to see. Pathologists usually look at slices, at samples that have been stained with hematoxylin and eosin, which have this pink and violet look. So, for example, but this is two-dimensional. In our three-dimensional bladder that we have acquired with light sheet microscopy, it doesn't look like this. It looks different because it's fluorescence, whereas this is a pigment. So it looks, for example, instead of pink and white, pink and violet on a white background, it looks red and green on a black background. It depends on where you store the channels. If I store one channel, one wavelength in the red channel and one other wavelength in the green channel, I get an image that looks red and green with a black background, like the ones that I showed you from the two-photon microscope. But then we apply a color transformation and we make these three-dimensional tomographies look like violet and pink with a white background. This is a simple, perhaps useless transformation. Probably not so, because anatomopathologists are used to look at samples that are stained like this. And since our fluorescent stains mark the very same structures that these pigments are marking, then we can create a three-dimensional view of the sample, in this case, a human bladder, but with the coloring of traditional microscopy, which is what the physicians want to have when making their diagnosis. And we are seeing that, for example, in some cases, if you have a tumor, for example, and even if you have a two-dimensional slice, but the tumor is so evident, so apparent, the diagnosis is simple. In other cases, we are working with physicians that would like to look at the sample in a more three-dimensional way, so that, for example, on edge cases where it's not clear if there's a tumor or not, by looking at the three-dimensional sample instead of a small slice, they should get a better diagnosis. So we are also working with, we are trying to bring this technology also to the clinical field. We're trying to see if there's margin for that.
More questions? So if not, let's thank Giacomo again.
Thank you.